Okay, so there's your picture of the first step of photosynthesis. You've got this. You've got the plant, the sun hanging out. It hits a leaf. We zoom into that leaf and say, well, what's going on? We zoom that leaf. It's got a cell. We know it's a eukaryotic cell because of the nucleus. We're looking at specifically the chloroplast. Inside the chloroplast, being a double membrane organelle, it's going to have the thylakoids. These are those little stacks, and then the liquid is the stroma, and the light waves will hit specifically the thylakoid. And in that thylakoid is the chloroplast. Okay? So let's actually write this out. Here we go. So, light shines on the leaf of a plant. That's like serious stuff right there, right? All right, so light shines on the leaf of the plant. We're going to get a little more technical than that. All right, so the light shines on the leaf of the plant, and then the light is then captured by what?
The cell.
By the chlorophyll. Yeah, I heard you. You're just rambling. In the thylakoid. So light is captured by the chlorophyll in the thylakoid. Okay, so we're using the sun's energy to form ATP. So what specific part of that plant cell are we going to use? What? No. The chlorophyll. Where's the chlorophyll located? What are these guys called? The thylakoid. All right, so these flattened sacs are called thylakoid. And the sun's energy is going to hit that thylakoid. On the top of the thylakoids is a chlorophyll. No. So again, the sun's energy, so this is light, is going to hit that thylakoid. On this thylakoid, there's little receptors, and those receptors are called chlorophyll. Okay, those are the pigments. So light's going to go into these thylakoids. What also do you have to get to a plant? Not only light.
Oxygen.
Not oxygen.
Carbon dioxide.
But we haven't got there yet, so hold on to that one. What's the other thing? So we got light, carbon dioxide, and what's the other one? Water. So we need to put water into this thylakoid. So water goes in to this thylakoid as well. Okay. So the light and the water interchange. And you should notice on that simulation that what kicks out when we mix light and water together?
Oxygen.
There you go. Oxygen. So O2 comes out. So light and water come in. The light actually splits the water in half. Because what's the chemical formula for water? H2O. So if we split water into its parts, what do we get out? 2 H's and 1 O, which is oxygen. And then when you take those hydrogens, we actually use them. We use those actually to carry electrons further on. So water goes in, light goes in, oxygen comes out. So one of the things that kicks out is what? ATP. So ATP goes out of the thylakoid. And also, the simulation just calls them electron carriers, but I do want you to know this. NADPH. Okay. And NADPH is an electron carrier. It carries electrons. Okay. Electrons, remember, electrons are units of energy in the chemical world. Okay, it's almost like cash. It's cash money, yes. No. All right, so the NADPH will carry those electrons. That's that extra hydrogen that's kicking out. And ATP is going to go into the Calvin cycle, but we'll look at the Calvin cycle in a little bit. So that's what's all going on in the thylakoid. So let's break this down. So what did I say the light does to the water? There you go. So light splits water releasing oxygen. So light's going to come in, it's going to split the water, it's going to release oxygen.
Do you know the reason?
When you do split water, this also produces... okay.
Carbon dioxide, I can't
ATP and H plus. This produces ATP and H plus. H plus is hydrogen. Okay? And H plus is carried by NADPH. And that's going to go to the Calvin cycle. So this produces ATP and H plus. H plus is carried by NADPH. And whenever you see H plus, you also need to think electrons because they go in alike. Because H plus is a proton, electrons are negative, so they actually go together to the Calvin cycle. So the NADPH, the ATP, the NADPH is going to carry the electrons. The ATP is going to allow energy for this to occur. It goes into what's called the Calvin cycle. Now the Calvin cycle is a multi-step cycle. Okay, there's a lot of different steps. But what you may need to know is that CO2 goes into the Calvin cycle. So CO2 goes into the Calvin cycle. And there's actually 6 of those CO2. So 6 CO2 go in the Calvin cycle. And more correctly, it's actually that the Calvin cycle goes through 6 times, and so it needs 6 CO2s. But since we're only going to draw this once, we're going to say 6 CO2s. Okay. Because what is why would we need 6 CO2s, not just one?
It doesn't have enough.
Yeah, you need a lot.
You need a lot, right?
Yeah.
Because what's the ultimate goal here? What are we going to make? What are we going to kick out here? Glucose. And what's the chemical formula for glucose? There you go. C what? C6. And so if we have 6 carbon dioxides, how many possible carbons do we have? 6. And if we have 6 carbon dioxides, so 2 oxygens for every 1 carbon dioxide, how many total oxygens do we have?
12.
12. So C6H12O6. So you actually end up using some of the oxygen to carry, but don't worry about that. But those C6 is from the 6 carbon dioxides. So you end up getting C6H12O6.
Oh my god.
Okay. So we need those 6 carbon dioxides to make 1 glucose. So it takes 6 turns of Krebs cycle to make 1 glucose. That's a lot. Right? So that's the reason why the plant can't just barely live off of, just a couple light beams. Every so often gives it light. No, it needs light all the time. Because if it doesn't have light, it can't capture the light to split the water. If it can't split the water, it can't release the hydrogens. If it can't release the hydrogens, it can't collect the CO2. So it's all connected.
Could we live without CO2?
Could we live without CO2? Yes.
What about plants?
No.
So we would die still, then we would have no
Technically, we couldn't, because then the plants would die.
without CO2. So no photosynthesis, and then we're gonna run out of trees, and there's gonna be no oxygen, and then we're gonna die.
We could just move on.
So
Yeah.
going back, okay, so this is a whole cycle. So ATP is used in the Calvin cycle, and then it kicks out ADP. The ADP is adding diphosphate. What's di mean?
Oh, wait, the color dye?
No, DI.
What is that, water?
Or duo, or dose, or duo. 2. So diphosphate means 2 phosphates. What's triphosphate mean then? 3 phosphates. So what it's going to do is it's going to lose a phosphate to allow the Calvin cycle to go on. It's going to kick that diphosphate back to the light reactions. And the light reactions are going to add another phosphate onto it. It's going to kick it back to the Calvin cycle. It's going to lose one of those phosphates. It's going to kick it back over the light reaction. It's going to gain a phosphate. It's going to keep on doing that. And the same thing with NADP. NADP is going to lose that hydrogen because it's going to lose an electron. And it's going to kick it back over as NADP. It's going to collect a hydrogen. It's going to become NADPH. It's going to kick it back over and keep on doing that. So this is a continual cycle going on. So that's all the Calvin cycle. There you go. Here's your words. So 6 CO2s are fixed in Calvin cycle. All right, so ATP. And NADPH are used and since they're used up, ADP and NADP are sent back to the thylakoid.
Does that say 1C?
What's that?
1C. Oh, no, it's sent.
Are sent back. So I skipped that box, right? Yeah. So 6 CO2s are fixed in carbon cycle. ATP and NADPH are used, and what are they used to? What do we make at the end? Yeah. To produce glucose. Okay, so we're going to use ATP, we're going to use NADPH, we're going to capture or fix that CO2, we're going to produce glucose. When we produce glucose, it's going to kick back the NADP and the ADP. And the main thing down here, this all occurs in our stroma. Okay, so it occurs in the stroma. So you do remember that it does occur in both plants and animal cells. So even though we have drawn our fancy fandangled plant cell, and we have our little nucleus, right, with the nucleolus in the middle. We have our gigantic vacuole, right, and we have our chloroplast. There are little lines in it. Okay, we have our chloroplast, which does what?
Turns stuff green.
Yes, because that's chlorophyll in it. But what's the process that occurs in the chloroplast? Photosynthesis. So not only do we have all that, we also have our mitochondria. I didn't draw this big enough, but and most time what you see is a little wavy thing in the mitochondria. And it's not shown on that picture, but it is a little wavy thing. And that wavy thing is the cristae. The vocabulary word y'all used today?
Yes.
Right? It's the folding of the inner membrane. Remember that mitochondria have 2 membranes? It's like if you have a young person and an old person, the old person's the inside. Why? Because the old person have? Oh, they have wrinkles.
That's me.
Right?
Awesome.
True though, right? So mitochondria essentially looks like a young person's skin on the outside of an old person's skin. Because the inside of the mitochondria has the cristae, and those are like little finger-looking foldies. And those foldies are very, very important, because if you don't have foldies, you don't have surface area. That surface area is very, very important. So not only do plant cells have mitochondria, so do fantabulous animal cells like us. And so what I was trying to draw was something like that. But that's my mitochondria. So they both do glycolysis. And glycolysis splits glucose. So glycolysis. That would be sugar. Glyco means sugar. Yeah. And lysis means to split. So that would be a sugar splitting. Sugar splits. Banana split. Kevin Bacon. What?
Mitochondria thing looks heavy.
Yeah. F. Right. So glycolysis is the entire purpose here is to split the glucose into actually two what's called pyruvates or pyruvic acids. So glucose, which is a 6-carbon sugar. Okay, there's 6 carbons in glucose. Splits into 2 3-carbon pyruvates.
Wait, what does it say there?
So glucose, parentheses, 6 carbons, splits into 2 3-carbon pyruvates. Pyruvate, Pyruvate. So we use 2 ATP, so we spend money, so we need to make some money now. So in the process of taking glucose and splitting it into 2 pyruvates, we actually do make, it makes 4 ATP. So it makes 4 ATP, which we call as a net gain. So it makes 4 ATP. Thus, this is a net gain of 2 ATP. So yeah, I know we spent 2 ATP, we gained 2 ATP. Call it even. So we make 4, we spent 2, so when I say net gain, that's basically how much we ended up with. So 4 minus 2 is 2. That's why the 2 ATP is there. And this, do it? No, we gained 2. Because we made 4, we spent 2, so we get 2 left over.
But you already had two. But if you wouldn't have spent it in the first place, then you wouldn't have came up with the same results.
Well, if you spin that 2 again to make 4, and then it makes 2 again, and then it does it.
So this does occur in the cytosol, more correct, which is the correct form. Y'all also always heard it as the cytoplasm. So this occurs outside of the mitochondria. So cytosol is the liquid that's in between the mitochondria and the cell membrane. So oxidative respiration creates ATP. Where does this occur? Where does respiration occur? In the what organelle? In the mitochondria. Good. So we're going to draw a really big mitochondria because we need lots of room. We're going to draw our wrinkled old man on the inside. What are those things called? Cristae. So these are the cristae. What's the inside called? David, what was that called? The inside? No. The inside. The matrix. Right?
It's like curving around.
Yes. Good job. Yeah. Okay, so there's your mitochondria. It is a double membrane organelle just like the chloroplast. Again, it has its own DNA, and what we assume because of endosymbiosis that it came from another organ. It was another organism at one time. So we got 2 pyruvates. And those 2 pyruvates want to go into the club. They want to go into the club. Yeah, and the club is mitochondria. And so when they're into the club, how do they get into the club? They have a secret passage. What do they pull in them with them? Almost. What do you breathe in? Oxygen. So their passage into the club is oxygen. So you notice up here on the top it says oxidative phosphorylation. That's oxygen. So to get into the club, it also pulls in some O2. Okay. If you don't have oxygen, you're not getting into the club. If you're not getting into the club, you're not going to get any energy.
So we go into the club.
Yeah.
No, Nathan, we're not going to the club. You need to do some research.
Once it's in the club, the mitochondria, it's going to go through the Krebs cycle. Because we already did Calvin, right? Calvin's old hat. All right, so this is called the Krebs cycle. Krebs. Kreb. So once we're in the club, we're going to go hang out in the cool room called the Krebs cycle room. All right? And it's kind of a cool little place, but we're not done yet. All right? So we haven't really made any ATP at this point. We're basically just getting in the club and getting settled. But there's a really cool room. It's actually a train. We're going to go through this little transport chain. And that chain goes into, I'm going to change color so you can see it. This extra part actually goes along this crystate. And we call this little train here the ETC. Get on the ETC! Yeah. Okay. So we're going to go from the outside as pyruvate. When we get into the club with what we need to bring in with us to allow us to get in? Oxygen. And we're going to go to the Krebs cycle. In the Krebs cycle, we're going to change around a little bit. We're actually going to collect a bunch of electrons. That's where the E comes from. And us and all our electron buddies are going to go down to the ETC. And we're going to go down along in the train, and we're going to make lots of ATP.
Yeah.
All right?
ATP and the ETP after.
Yeah, you make a bunch of ATP after you get back out of the ETC. And we're like, yeah, we got ATP, we got some energy. Yeah. And then we go use those energies to go do other cool things like, I don't know, like breathe and weird things like that. All right, so we got our pyruvate, we got some oxygen. So we're going to do this. So again, pyruvate with what? How are we going to get into the club?
Oxygen.
Oxygen. With O2. Goes through Krebs.
Was that pyruvate?
Pyruvate. Krebs cycle. So pyruvate goes with O2, goes through the Krebs cycle.
I thought it was Krebs.
Yeah, Krebs. Krebs.
B or B?
B. B as in boy.
Oh, I thought it was
It's not. Yeah, it's not Russian. The Krebs cycle. So when we were in the club at the Krebs cycle, we found some bad stuff in there. So they got kicked out. And we breathe that out. What do we breathe out?
O2. See, I mean, carbon dioxide.
Carbon dioxide.
O2.
So it produces CO2. But there's also some other things. You know, I told you, what do we pick up in the Krebs cycle? A bunch of what?
ATP.
No, we haven't made ATP yet.
I mean, not the Krebs cycle.
Electrons. Now these electrons, they kind of hang out by themselves, right? So they got to have a carrier. And what was our carrier last time to carry the NADPH? Okay, so the carriers here are a little different because this is a different place. So we actually use what's called NADH without the P. So NADH and FADH2. What? So those guys up here on the top, so these NADHs and FADH2s, they carry electrons to the ETC. And I would E minus, which is electron. And I would make sure I write all ETC as electron transport chain. So you make sure you know what that is. I just put ETC because I don't have enough room. Say that again. So the electrons, and a lot of times it's electrons or hydrogens. We use them interchangeably. So I'm going to also put electrons / hydrogens because it's actually more correct. The hydrogens are pumped through something called the ATP synthetase. S-Y-N S-Y-N-T-H-E-T-A-S-E. ATP synthetase. S-Y-N-T-H-E-T-A-S-E. And this makes 32 ATP. It's really small on the bottom. But what it says is it says electrons or the hydrogens are pumped through the ATP synthetase, and this makes 32 ATP. So remember, we only spent 2 to start all this, right? How many do we get out?
32.
32 plus another 2, so we get between 34 to 36 each time we go through this cycle. That's a lot of ATP. That's a lot of money. Okay, so we're lifting dumbbells, we're doing lots of breathing, we're running out of oxygen. What happens to our body? It gets tired, right? We start sweating, we start breathing hard, and our muscles actually start to cramp up, right? Because of lactic acid fermentation. Okay, so those are muscles. Okay, so I'm going to go down here to this box right here. So without, that's WO, without, right? Without oxygen, pyruvate produces either lactic acid in the muscles. Okay, so without O2, pyruvate produces lactic acid in muscles, and that's that pain, stickiness that hurts your muscles a lot. Or if you're a grape grower and you pull the, that's my grapes. It's grapes, yeah. Duh. Obviously those are grapes, clearly. And you pull all the oxygen out and you have yeast in there, what does it make? Almost. Can't drink this grape juice. It makes what? Wine, right? So pyruvate either produces lactic acid if it's in muscles, or it'll make what's called ethyl alcohol. Okay. The other one's called ethyl alcohol and CO2. Okay. So if there's no oxygen, pyruvate produces either lactic acid in our muscles, or if you're yeast, or a lot of times if you're well, bacteria produce lactic acid as well. But if you're a yeast, you'll produce ethyl alcohol, and ethyl alcohol is what makes alcohol, the wine. Okay. Okay, so you got all that? We good? Okay, so we need to change gears real quick because we're going to talk about something that's kind of related to this, not really. But you ever heard of this idea of stem cells? Stem cells? Okay, one of the things you're supposed to write down today on vocab was differentiated and stem cells, right? What does it mean for a cell to be differentiated?
It means